The sweat leaving your skin right now has almost no smell. The smell arrives later. A droplet can emerge as a dilute solution of water, sodium, chloride, lactate, urea, and trace molecules, then sit on the surface while organisms too small to see begin taking it apart. What another person eventually detects is not simply your sweat. It is a chemical afterimage produced by your glands, your genes, the microbes living in a particular fold of skin, the fabric against that skin, and the nose receiving the signal. Two people can run the same distance in the same heat, lose nearly the same mass of water, and smell completely different afterward. One may produce almost no recognizable underarm odor. The other may release a sulfurous note at a concentration measured in parts per trillion. The difference is not a verdict on cleanliness. It is a reaction pathway. Your body supplies ingredients that are mostly non-volatile. Bacteria perform the final chemistry. Air carries the products away. A nose converts those airborne molecules into electrical signals. What we call body odor is therefore not one substance and not one event. It is a collaboration. Begin with the part of sweating that has nothing to do with smell. Humans possess roughly 2 to 4 million eccrine sweat glands, distributed across nearly the entire skin surface. Lindsay Baker's 2019 synthesis of sweat gland physiology reported densities of about 250 to 550 glands per square centimeter on the palms and soles, with lower densities across the trunk and limbs. Each gland is a narrow tube, several millimeters long, with a secretory coil buried in the dermis, and a duct rising toward the surface. When thermoreceptors detect increasing heat, the preoptic region of the hypothalamus drives sympathetic nerves to release acetylcholine. That detail is unusual. Most sympathetic nerves use noradrenaline, but the fibers controlling eccrine glands are mainly cholinergic. Acetylcholine binds muscarinic M3 receptors on secretory cells. Calcium-dependent ion transport follows. Chloride moves into the glandular lumen, sodium follows the electrical gradient, and water follows the osmotic gradient through pathways that include aquaporin 5. A primary fluid forms deep inside the coil. That first fluid is closer to plasma, in salt concentration, than the droplet you finally see. As it climbs the duct, epithelial sodium channels and the cystic fibrosis transmembrane conductance regulator help recover sodium and chloride. The duct is relatively impermeable to water, so the emerging sweat is usually hypotonic, more diluted than blood. In cystic fibrosis, defective CFTR reduces chloride reabsorption and produces unusually salty sweat, the basis of the clinical sweat chloride test. In everyone else, the system adjusts with flow. When sweat moves slowly, the duct has more time to reclaim salt. When it rushes, more sodium and chloride reach the skin. The purpose is thermal, not detoxification. Evaporation consumes energy because liquid water must absorb latent heat to become vapor. At skin temperature, evaporating 1 gram of water removes roughly 2.4 kilojoules of heat. A liter that completely evaporates can therefore carry away about 2.4 megajoules. Sweat that drips to the floor has spent water without delivering the full cooling return. Humidity matters because the atmosphere can accept only so much additional vapor. None of this requires an odor. Eccrine sweat is overwhelmingly water and electrolytes. It can contribute lactate, amino acids, urea, and moisture that changes the local habitat, but the familiar underarm scent is driven largely by another gland. Apocrine glands cluster in the axilla, around the nipples, and in parts of the anogenital region. They are larger than eccrine glands, become functionally active around puberty, and empty into hair follicles rather than directly onto the surface. Their secretion is richer in lipids, proteins, steroids, and molecular precursors that bacteria can use. A third gland type, the apocrine gland, develops in the axilla during adolescence and releases abundant watery fluid directly onto the skin. One armpit is therefore not a single faucet. It is a mixed chemical landscape, supplied by eccrine, apocrine, apocrine, and sebaceous secretions, plus shed cells and residues from everything placed on the skin. This distinction explains an observation that seems contradictory until the anatomy is separated. A person can sweat heavily with little odor because high-volume eccrine output is not identical to high precursor output. Another person can produce a modest amount of visible moisture, yet develop a strong scent because apocrine precursors and odor-forming microbes are present in the right combination. Amount changes transport and humidity, Composition changes chemistry. They interact, but they are not the same variable. Palms reveal the split. They contain some of the highest eccrine gland densities on the body and can become visibly wet during heat or emotion, yet they lack the apocrine architecture of the axilla. A sweaty palm does not normally develop the classic underarm note. Location changes the machinery. The axilla adds the next mechanism. It is warm, folded, humid, partly shielded from airflow, rich in hair follicles, and repeatedly supplied with organic material. Those conditions create a microbial habitat. Common residents include species of Staphylococcus, Corynebacterium, Cutibacterium, Anaerococcus, and Peptoniphilus. Calling them simply bacteria that eat sweat is directionally useful but chemically incomplete. Different species possess different transport proteins and different enzymes. One organism may take up a precursor and release a volatile sulfur compound. Another may split an amino acid conjugate to release a branched fatty acid. A third may occupy space without producing much recognizable odor at all. The community is less like a layer of dirt and more like an industrial district in which neighboring factories own different machines. Dermatologists Walter Shelley, Herbert Hurley, and Arthur Nichols isolated the logic in a landmark 1953 paper in the Archives of Dermatology and Syphilology. Fresh apocrine material did not carry the mature odor by itself. Odor developed when bacterial activity acted on glandular secretion, and suppressing that activity interrupted the process. Their analytical tools were primitive compared with modern mass spectrometry, yet the core conclusion survived 7 decades of molecular work. The human gland does not need to emit a ready-made cloud of malodor. It can release quiet precursor molecules. Time and microbes do the rest. This is why washing can temporarily reset the surface even though it does not alter the genes or remove the glands, and why odor returns at different rates in different people. The substrate supply resumes. The community rebuilds. The reaction restarts. In 1991, Andrew Layden and colleagues examined the axillary flora of 34 men and compared microbial populations with judged odor intensity. The density of aerobic coroniform bacteria was associated with stronger odor, with a reported Spearman probability of 0.001, and communities dominated by those organisms were more likely to be highly odorous. That did not mean every Corynebacterium strain produced the same compounds, or that Staphylococci were irrelevant. It showed that head count alone was not enough. Identity mattered. Later DNA-based surveys revealed even greater interpersonal variation than culture plates had captured. 2 armpits can have similar total bacterial loads, but very different metabolic capacity, because the decisive genes may exist in only a narrow branch of the community. The community also changes across distances smaller than a fingertip. Oxygen is more available at the exposed edge of the axilla than deep inside an occluded follicle. Water activity rises after sweating and falls as the surface dries. Salt becomes more concentrated during evaporation. Skin normally maintains an acidic surface, often called the acid mantle, but axillary pH can sit higher than more exposed regions because sweat, occlusion, and product residues continually reshape it. Enzymes have pH ranges in which their electrical charges and 3-dimensional structures work best. A shift of 1 pH unit represents a 10-fold change in hydrogen ion activity. That can alter bacterial growth, precursor transport, and enzyme speed at the same time. The consequence is not a simple rule that lower pH always means no smell. It is a change in which organisms and reactions are favored. Competition matters as much as growth. Staphylococcus epidermidis can occupy space and consume resources without carrying the specialized ShPatB route that releases 3M3SH. If it dominates, it may dilute the functional influence of rarer odor producers. If a product, antibiotic, climate change, or new routine suppresses one population more than another, the odor profile may change before the total number of bacteria visibly changes. Modern microbiome sequencing measures who is present by reading marker genes, but presence alone does not prove that a metabolic gene is active. Metatranscriptomics can measure RNA, and metabolomics can measure chemical products, yet relatively few axillary studies combine all three. The map is improving. It is not complete. Now the pathway becomes molecular. One of the most powerful underarm odorants is 3-methyl-3-sulfonyl-hexane-1-ol, shortened to 3M3SH. It belongs to a family of thioalcohols, compounds containing sulfur and alcohol groups. Human noses can detect certain sulfur odorants at astonishingly low airborne concentrations, so abundance is not the same as impact. Volatile fatty acids may be present in larger quantities, while trace thioalcohols can dominate the recognizable sulfurous character. The apocrine gland does not simply pour free 3M3SH onto the skin. It delivers an odorless conjugated precursor, in which the future odorant is chemically attached to a cysteine glycine dipeptide. The odor is locked inside a larger, non-volatile package. Staphylococcus hominis can unlock it. In 2018, Dilek Minhas, Daniel Bawden, Gavin Thomas, and colleagues used X-ray crystallography and biochemical assays to characterize a membrane protein in S. hominis. The protein, now called PEP-TSH, or DTPT, in this pathway, recognizes the modified cysteine glycine precursor and imports it by coupling transport to an inward proton gradient. It is a molecular turnstile powered not by a wire or battery, but by unequal proton concentration across a bacterial membrane. Once the precursor enters, an aminopeptidase named PEP-A removes the terminal glycine. The remaining cysteine-linked molecule is still not the final odorant. One more bond must break. Michelle Rudden, Raim Herman, Gavin Thomas, and their collaborators identified that final machine in a 2020 Scientific Reports study. The enzyme is a pyridoxal phosphate-dependent cysteine thiol lyase called ShPatB. Pyridoxal phosphate is the active form of vitamin B6, a chemical cofactor used by many enzymes to rearrange amino acids. ShPatB holds the precursor in a narrow hydrophobic pocket and cleaves the carbon-sulfur bond, releasing volatile 3M3SH, along with pyruvate and ammonia. The researchers solved crystal structures down to 1.6 angstroms for ShPatB and 1.4 angstroms for an inhibitor-bound form. At that scale, the binding pocket is no longer a metaphor, it is a measured arrangement of residues. In kinetic tests, ShPatB's catalytic efficiency toward the odor precursor was 138 times its efficiency toward cystathionine, a more conventional sulfur-containing substrate. The decisive experiment was genetic. The team placed the ShPatB gene into a Staphylococcus aureus strain that did not normally produce this thioalcohol. The modified bacterium gained the ability to release 3M3SH. One gene supplied the missing function. Within that tested system, ShPatB was necessary and sufficient for thioalcohol formation. The dominant skin-resident Staphylococcus epidermidis does not normally perform this transformation, even though it can be abundant. A lower abundance population of S. hominis can therefore have an outsized effect if it carries the right enzyme. This is one reason two people with equally many bacteria can smell different. Metabolic function is concentrated, not democratic. There is a parallel branch. Corynebacteria can release odoriferous short-branched acids, including 3-hydroxy-3-methylhexanoic acid, usually shortened to OSMA, from glutamine-linked precursors. A bacterial aminoacylase performs the cleavage. MHA has been described as pungent, sweaty, or cumin-like, while 3M3SH can read as sulfurous, onion-like, or tropical depending on concentration and context. Other volatile acids add sour, cheesy, or rancid notes. Steroid derivatives may contribute musky dimensions, though their quantitative importance varies, and human perception of individual steroids differs sharply. What reaches a nose is a blend. Change the ratio of thioalcohols to volatile acids, and the character changes, even if the overall intensity does not. Change which enantiomer, or mirror image form, is present, and receptor binding may change again. Human odor is a chemical chord, not a single note. Volatility is what turns local chemistry into a signal at a distance. A precursor can be abundant, yet remain effectively silent, because it stays dissolved in the wet film or bound to an amino acid. Cleavage reduces its chemical restraint. The product can partition from the skin into the air, and body heat increases molecular motion. Air currents then determine whether it accumulates under clothing or disperses. Odor impact depends on concentration divided by sensory threshold, not concentration alone. A trace compound with an exceptionally low threshold can outweigh a much more abundant molecule that receptors detect poorly. This is why a chemical analysis listing only the largest peaks can miss the molecule that defines the human experience. Analytical chemists combine gas chromatography, which separates volatile molecules in time, with mass spectrometry, which fragments them by mass-to-charge ratio, and trained sensory panels, which reveal which separated peaks actually carry recognizable odor. Instrument and nose answer different questions. The sulfur pathway also gives the bacterium a reason to participate. When S. hominis processes the cysteine-glycine precursor, it can recover glycine, pyruvate, and ammonia, usable carbon and nitrogen chemistry, while 3M3SH leaves as a byproduct. The microbe is not attempting to embarrass its host. It is harvesting a nutrient. Rudden and colleagues' phylogenetic analysis suggested that the specialized ShPatB enzyme entered an ancestral staphylococcal lineage through horizontal gene transfer roughly 60 million years ago, though evolutionary dates at that depth carry uncertainty. The enzyme's hydrophobic pocket then became unusually well suited to the branched human precursor. Host and microbe now meet through a pathway older than Homo sapiens. The host controls the first half of this supply chain. An ATP-powered transporter called ABCC11 sits in apocrine secretory cells and helps move odorant precursors into glandular secretions. A single DNA letter change at position RS17822931 replaces glycine with arginine at amino acid position 180. The G version supports functional transporter expression. The A version destabilizes the protein. In people with 2A copies, much less functional ABCC11 reaches the relevant cellular location. The downstream consequence is striking. Amino acid conjugates that would become characteristic axillary odorants are greatly reduced or absent. The bacteria can own the correct machinery and still receive very little substrate. The same variant controls a seemingly unrelated trait, earwax. Ceruminous glands in the ear canal are modified apocrine glands. Yoshiura and colleagues reported in Nature Genetics in 2006 that the AA genotype is associated with dry earwax, while GA and GG are associated with wet earwax. 3 years later, a study of 79 Japanese patients who had sought surgery for axillary osmidrosis found that 78 carried GG or GA. Among 161 people from the general Japanese population used for comparison, 57 carried those genotypes. The association was extremely strong. The link is anatomical rather than mystical. Ear canal and underarm glands use overlapping secretory machinery, so one transporter variant leaves evidence in both places. The result is not a binary switch between smell and no smell. Mark Harker and colleagues examined ABCC11 genotype, axillary headspace chemistry, skin metabolites, microbiota, and hygiene behavior in a 2014 Journal of Dermatological Science study. Participants with AA had substantially lower odor and lacked specific volatile odorants detected in carriers of the G allele, but trained perception still found appreciable odor, and all genotype groups produced odoriferous short-chain fatty acids. ABCC11 strongly changes the precursor landscape. It does not erase every route to scent. Eccrine material, sebum, other apocrine compounds, microbial fatty acid metabolism, clothing, and environmental molecules remain. Hormones help determine when that landscape becomes active. Apocrine glands exist before adolescence, but their secretory behavior changes with puberty, when androgen signaling remodels gland activity and sebaceous output. This is why a child can lose large volumes of eccrine sweat during play without producing the same axillary blend that appears a few years later. Adult differences often get reduced to sex, but the chain contains too many overlapping variables for a single label to predict an individual. Androgen exposure, gland size, hair, skin physiology, products, clothing, and microbial composition can all influence the result. Studies may report group averages while distributions overlap broadly. A group difference does not tell you which pathway dominates in the person standing beside you. Within one person, the two axillae are not guaranteed to be chemical twins. Product application can differ by hand dominance. Hair density, friction, clothing pressure, and small skin injuries can differ side to side. Microbial colonization begins from chance events and repeated contact. Researchers sometimes exploit this by using one axilla as a treated site and the other as a control, a split-body design that reduces genetic and dietary noise. The design itself carries a lesson. Even when the host genome is held perfectly constant, a local intervention can change odor. Genes load the supply system. Local ecology decides what happens next. This is also where careless statements about ethnicity fail. The frequency of Rs1782291 varies among populations, and the A allele reaches very high frequencies in some East Asian populations. That is a population genetic pattern, not a rule that identifies an individual. George Preddy's group used gas chromatography and mass spectrometry to compare 30 men of African American, European American, and East Asian ancestry. They found quantitative differences in volatile compounds and a strong role for ABCC11, but concluded that the genotype did not explain all variation. Every population contains biological diversity. Diet, age, products, microbial ecology, and other genes operate inside every ancestry group. A DNA test identifies a genotype. A label does not. Even identical precursor supply would not produce identical odor because the axillary microbiome is unstable enough to respond to the environment and stable enough to preserve personal differences. Skin pH, moisture, salt concentration, oxygen, hair, friction, immune peptides, washing practices, and cosmetic ingredients all apply selective pressure. Corynebacteria tend to thrive in moist regions. Staphylococci tolerate salt well. Hair increases surface area and can retain secretion, microbes, and volatile compounds, but shaving also perturbs the habitat and can change product contact. None of these variables operates alone. A humid fold under occlusive fabric is a miniature climate system. Julie Urban and colleagues at North Carolina institutions followed 17 people for 8 days in a 2016 Peer study. Participants were habitual antiperspirant users, deodorant users, or users of neither. The researchers first sampled normal routines, then had everyone stop products, then had everyone use antiperspirant. When products stopped, culturable bacterial density rose toward the level found in habitual non-users. When antiperspirant was applied, density fell sharply. The community composition also differed. People who normally used no product were more often dominated by Corynebacterium, while participants who had used deodorant or antiperspirant and then stopped showed more Staphylococcaceae. The sample was small, so it cannot define a universally healthy armpit microbiome. It does demonstrate that a hygiene product is not merely perfume laid over a fixed ecosystem. It changes the ecosystem being perfumed. Deodorant and antiperspirant solve different parts of the equation. A deodorant may mask odor, lower pH, or inhibit microbial growth. An antiperspirant reduces fluid delivery by forming temporary plugs in eccrine ducts with aluminum salts, especially when applied to dry skin, where the chemistry can occur before heavy flow dilutes the product. Less moisture changes transport, humidity, and microbial activity. Neither product rewrites ABCC11. Neither permanently removes S. hominis. Their success depends on the dominant route in a particular person. A fragrance-only product can fail when rapid bacterial conversion continues beneath it. An antiperspirant can reduce wetness, while leaving some apocrine precursor chemistry intact. A broad antimicrobial can suppress odor yet also rearrange competition among resident species. The underarm returns to chemistry, not morality. Aluminum salts illustrate the difference between suppressing flow and neutralizing molecules already formed. In water, they hydrolyze and form polymeric species that combine with material in the upper sweat duct, narrowing the route temporarily. The effect fades as normal skin turnover and washing remove the plug. An acidifying deodorant attacks a different variable by shifting surface pH. An antimicrobial attacks cell growth or membrane integrity. An enzyme-targeted compound would be narrower still. It could, in principle, block DTPT uptake or the ShPatB active site while leaving much of the surrounding community intact. The 2018 transporter structure and the 2020 lyase structure make that strategy chemically imaginable. Whether a candidate remains safe, stable, effective in a real axilla, and selective over repeated use requires clinical evidence. A beautiful binding pocket is a target, not yet a product. Timing follows mechanism. Applying antiperspirant to a dry surface before sleep can allow duct plugs to form during a period of relatively low sweat output. Applying it to an already wet axilla creates dilution and can increase irritation. Washing removes accumulated volatiles, precursors, cells, and some microbes, but recolonization begins immediately from follicles and neighboring skin. Fragrance changes the mixture reaching the nose, without necessarily reducing the source. These are not interchangeable actions. One reduces fluid, one reduces biomass or reaction rate, one removes products, one adds competing odorants. Fabric becomes an external organ of that chemistry. In a 2014 applied and environmental microbiology study, Chris Callowert and colleagues collected shirts worn during a fitness session and compared cotton with polyester. Trained odor assessors judged polyester shirts more intensely odorous and less pleasant. Microbial analysis showed that Micrococcus species grew better on polyester in the experimental conditions, while the starting axillary communities on skin were more dominated by Staphylococcus and Corynebacterium. Synthetic fibers are hydrophobic and interact differently with oily volatile compounds than cellulose fibers do. They can retain residues through washing, then release them again when rewarmed and re-wetted. The odor that seems to return minutes after dressing may not have been generated entirely on the skin that morning. Some of its substrate, microbes or volatiles, were waiting in the garment. Stress adds another route into the same chamber. Thermal sweating is coordinated to protect core temperature, but emotional sweating can rise rapidly in the palms, soles, face, and axilla. In studies using the Trier social stress test, in which participants face public speaking and mental arithmetic under evaluation, heart rate and salivary cortisol rise alongside axillary sweat. The chemistry produced during a tense meeting can therefore differ in timing and glandular mixture from the chemistry produced during a steady run. Faster wetting changes diffusion. Apocrine secretion may add richer substrates. Fabric remains close to the skin. A person who says stress sweat smells different is not necessarily imagining a new odor. The glandular inputs and environmental conditions really can be different, although assigning a single compound uniquely to anxiety would overstate the evidence. Diet can modify body odor, but its effects are specific rather than a vague process of sweating out toxins. Volatile molecules from garlic can enter breath and skin emissions after metabolism. Alcohol can alter emitted volatiles directly and change heat loss and sweating. In a controlled crossover study by Jan Havlicek and Pavlina Lenichova, 17 male odor donors ate either meat or no meat for 2 weeks, wore axillary pads during the final 24 hours, then crossed to the other diet. Female raters judged odor from the non-meat condition as more attractive, more pleasant, and less intense. That small experiment measured perception, not a universal molecular law, and it cannot show that one meal permanently changes someone's baseline scent. It does show that substrate supply extends beyond the sweat gland. Metabolism leaves volatile traces. Hydration illustrates another common misunderstanding. Drinking water can affect total fluid balance and, during heat, the ability to sustain sweating. It does not rinse apocrine ducts from the inside like water through a pipe. The kidney tightly regulates plasma composition, eccrine ducts reabsorb ions, and apocrine precursor transport uses specific cellular machinery. Likewise, sweating in a sauna is not a major detoxification route. Baker's physiological review concluded that kidney and gastrointestinal excretion dominate removal of most waste intoxicants, while sweat measurements are easily distorted by contamination from skin. More water can change concentration under some conditions. It does not switch off the microbial enzymes that create 3M3SH. Age changes the system again. Apocrine activity becomes prominent at puberty under hormonal influence, helping explain why the familiar adult axillary profile is uncommon in young children, even though children already sweat eccrinely. Later in life, gland output, skin lipids, hormones, medication, and microbial ecology shift. In 2001, Shinichiro Hayes and colleagues analyzed body odor from people aged 26 to 75 using headspace gas chromatography and mass spectrometry. They detected 2-nonenal, an unsaturated aldehyde with greasy and grassy qualities, only in participants aged 40 or older in that sample. Omega-7 unsaturated fatty acids and lipid peroxides increased with age and correlated with 2-nonenal. Oxidative degradation experiments generated the compound from those lipids. That finding established a plausible pathway for one age-associated note. It did not prove that everyone over 40 produces it, or that one molecule defines the smell of aging. Health can sometimes produce a qualitatively different signal, but ordinary underarm odor should not be turned into a diagnosis. In trimethylaminuria, variants affecting the liver enzyme flavin-containing monooxygenase 3 can reduce conversion of fishy trimethylamine into the much less odorous trimethylamine N-oxide. Trimethylamine can then leave through breath, urine, and sweat. That is a systemic metabolic pathway, not the standard axillary thioalcohol route, and it is rare. Some infections, medications, and metabolic states can also alter odor or sweating. A sudden, persistent change accompanied by other symptoms deserves clinical evaluation. Smell alone cannot distinguish a bacterial shift from a drug effect, a garment, a dietary exposure, or a metabolic disorder. The chemistry contains information, but the codebook is incomplete. There is one final source of variation, and it sits in the observer. An odor molecule does not carry a fixed adjective. It binds to a pattern of olfactory receptors whose genes vary among people. Then the brain interprets that pattern through concentration, mixture, adaptation, memory, and context. One person may be highly sensitive to a steroidal component that another barely detects. At a low concentration, a sulfur-containing molecule can add a subtle savory or tropical edge. At a higher concentration, the same receptor pattern may become piercing. Continued exposure produces adaptation, which is why a person may stop noticing their own background odor while someone entering the room detects it immediately. The producer varies. The receiver varies too. Now the layers converge. Visible sweat begins with neural control, ion channels, osmotic water flow, and heat transfer. Recognizable underarm odor begins with a different distribution of glands and a richer set of non-volatile precursors. ABCC11 controls how much of a major precursor class enters the secretion. Microbial transporters pull selected molecules into selected cells. PEP-A trims a peptide. ShPatB breaks a carbon-sulfur bond. Rhinid bacterial enzymes release branched acids through a parallel path. Humidity sets reaction conditions. Products shift water and microbial abundance. Fabric stores and re-emits the mixture. Diet and age alter available molecules. A genetically different nose converts the final cloud into experience. This is why more sweat does not map cleanly onto more smell. It is why identical twins can drift apart after adopting different routines and environments, yet strangers with similar ABCC11 function and microbial pathways can produce related chemical notes. It is why washing works but does not last forever, why a shirt can smell stronger than the person who wore it, why puberty changes odor without inventing sweat glands from nothing, and why dry earwax can reveal something about underarm precursor transport. Each observation belongs to a different layer of the same mechanism. Return to the first droplet. It emerged to move heat, carrying almost none of the odor that would later define it. On the skin, it met a private ecosystem, received molecules from neighboring glands, entered bacterial membranes through proton-powered gates, and surrendered bonds to enzymes shaped by millions of years of evolution. Some products escaped into cotton, others clung to polyester. A few reached another person at concentrations so low that no human eye could ever see the amount involved. The scent was not stored inside the water. It was assembled across bodies, genes, microbes, fabric, air, and perception. You do not have a smell, you have a living reaction.